Cover measurement methods using points and lines. As we learned in the previous lesson, there are a variety of different approaches that we can use to measure plant cover. And in this lesson, we'll be focusing on methods that use points and lines. Point-intercept methods are one of the most commonly used methods to estimate cover. We can use single points, points that are arranged on transects, in point frames, or on point quadrats. We record the number of hits on canopy, foliage, basal vegetation, or soil surface, depending on the type of cover that we're trying to estimate. And the calculations are quite straightforward. The percent cover equals the number of hits on a particular species divided by the total number of points that were measured. We multiply by 100 to convert this to percent cover. In the example that we see in front of us, there are 8 different points that were measured along a transect. And this is a very simple system in which there's a grass and a forb. If we look at how cover is estimated from this example, we see that there were 5 of those points that hit the grass. So 5 out of 8 multiplied by 100 equals 62.5%. For the forb, there was only one example where the point hit the forb species, and even though it's hit 2 different leaves on that forb, it only counts as one hit, which equals 12.5%. Let's look at a slightly more complicated example. This shows a stand of vegetation with different kinds of ground cover, and we're going to run a transect with 10 points. We are recording multiple types of cover, and so that includes ground cover, basal cover, foliar cover, and canopy cover. We can see the data that was recorded for each one of the points. The first point did not hit anything. In the second point, there were no hits on any plant materials, so basal cover, foliar cover, and canopy cover do not have any hits, but there was one hit on the rock for the ground cover category, so that is recorded as rock. For the next point, in this case, the point hits a leaf, and so it's recorded as foliar hit, and also because foliar hits are included in the canopy of the plant, it's also recorded as a canopy hit on grass. In this case, there was no ground cover recorded because it hit bare soil. We can see by examining all of these points and then totaling them up that we had 4 hits in ground cover for 40%, 1 hit on basal cover here on point 5 for 10%, and that the foliar hits, or the foliar cover, at 20% is less than the canopy cover at 50%. And this makes sense because foliar cover is always less than the canopy cover because foliar is included in canopy. So let's look at another approach, which is a step point transect. This is also called a pace transect. In this case, we determine to walk in a specified direction, and we take measurements at regular intervals where we record the hits. The way this is actually done is that as someone is walking along at the specified time when they pace that they are supposed to take a point, they will usually raise their foot to a 30-degree angle and then drop a pin flag at the tip of their shoe or boot, and then record what that hits. This approach is very good for monitoring because it is rapid. It does require a large number of points, usually between 300 to 500 points per area. And one of the problems with step point transects is that they are subject to bias because of where the pin is lowered, and also the tendency for someone who's walking to try to avoid walking through plants that are particularly nasty due to being spiny or in some other way undesirable. So let's look at how we can take multiple measurements. Remember, we have canopy cover and also ground cover, and so we measure oftentimes multiple types of cover at the same time. Also, we can hit more than one species, and we want to take measurements there. So in this case, we're lowering a pin flag, and I would like to mention at this point that we do want to usually use a pin flag or something with a very small diameter, because a point in theory does not have any dimension at all. And with this first pin flag, it hits the bare grass twice, it hits blue grama once, and it hits a globe mallow before it lands on a rock. With the second pin flag, this point hits the bare grass, and then it hits soil or bare ground. So let's look at this a little more closely on how we would record this. For point number 1, because the top canopy hit was on bare grass, that is recorded here. Now, the lower canopy layers, if there are hits on other species in lower layers, there's room for that to be recorded as well. And we record blue grama and globe mallow because those were hit subsequently to the top canopy hit. Note that we don't write bare grass even though we hit it twice, because you only record one species once for each hit. And then finally, at the soil surface, the rock is recorded up here. For the second point, the hit for top canopy layer was on bare grass, and then there were no lower canopy hits, and the soil surface hit was on bare ground or soil. If we look at a slightly more complicated system, again, this has just 2 species. We're going to look at a series of hits. The first one was a miss. The second one, there is a top and a sub-canopy hit, so 2 species. The third one, there's a top canopy hit. The fourth one, a top canopy and sub-canopy hit. And on this fifth one, there's a top canopy hit and also a basal hit, or hit on the base of the plant. And this table shows how these data would be recorded. Let's take a look at how we would then analyze this for ground cover and for canopy cover. For ground cover, there were 2 hits out of 5 on rock for 40% cover of rock. There was 1 hit on a basal vegetation out of 5 for 20%. And the total ground cover then is the recordings of all of the ground cover hits divided by the number of points that were taken, or 60%. Now you may wonder why soil doesn't show up in our ground cover classifications, and that's because remember that bare ground or bare soil is not a ground cover category. Ground cover is defined as those features that cover soil. Okay, now if we look at canopy cover, we sum for each individual species the number of hits. So for blue grama, there were 4 hits out of 5 for 80%. cover. So we sum the top layer and lower layers for our canopy cover categories. Similarly with Globe Mallow, there was one top canopy hit and one lower canopy or sub-canopy hit for 2 hits, or 40%. Now you may note that the total canopy cover equals 80, not the sum of these two, which would be 120%. And the reason for that is that when we are recording or calculating total canopy cover, we only use top layer hits. If we were to include lower canopy hits, then that would be artificially increasing the number of hits on the top canopy. And we would get a value that is an inflated value for the total canopy cover. So let's discuss how we place our points throughout the landscape. Well, one of the intuitive ways to do it is to randomly place single points throughout the study area. And while random placement is intuitively appealing, it's not very practical, because in order for canopy estimations or cover estimations to work with points, we usually need a large number of points. If we went to each one of these points, it would take time to go to each point, and also we would only get one measurement per point. So in general, this is not an approach that we take for placing points in the environment. We have seen that it is very common to place points along transect lines. We also can place points on quadrats. So if we're doing measurements that use quadrats, we can measure cover at various locations, not necessarily all of these locations, sometimes just one or two on a quadrat frame. Another approach is to use grid point frames, and in this case we have a frame that is fitted with crosshairs, usually made out of some sort of filamentous material such as string or wire or fish line. And there's usually a pair of the frames that are held together in one solid configuration. We see a picture here of someone using a pin flag. So the way that grid point frames work is that we measure we take a measurement of cover at each one of the intersections of these points in the grid. And so by having the two frames superimposed over each other, then we have very specific control on the angle of placement of those pins. In this case, the grid point frame is the sampling unit, and each one of the point measurements is a subsample. Another option is to use a pin frame. In this diagram we see a 5-pin frame, and the way that this works is that the pin frame is the sampling unit, and each pin is lowered and it's recorded what kind of canopy and also what the ground surface cover is where that pin is lowered. Here we see a picture of a pin frame. In this case, this has 10 points or 10 pins. Each one is lowered into the vegetation. And we can see from these diagrams that pin frames generally are best used in relatively dense and somewhat homogeneous vegetation. Pin frames do not work very well in sparsely distributed vegetation. Another advantage of pin frames is that they can be made so that you can angle the point. So rather than lowering the point vertically, we can actually lower the point at a specific angle, and sometimes this is a desirable trait. So you may be wondering, what is the appropriate number of points to be taken? So far we've looked at examples that have all been based on very few points, and that by just adding 1 or 2 points you can have a very remarkable effect on the estimate of cover. The number of points that we collect depends on our management and our sampling objectives, why we're collecting the data, and how we plan to use the data. It also depends on the system that we're working in, the site characteristics, as well as the characteristics of the species that we're working with. In very sparse vegetation, you could imagine if you had a plant species that had only 1% cover, on average we would have to measure 100 points just to detect that plant once. And so the number of points is definitely affected by the kind of vegetation. The type of point measurement method that we're using is also very important, as well as the kind of sampling unit. Are we measuring points along transects? If so, how many points per transect and how many transects? Same thing with using point frames. How many points per frame and how many frames? As we've discussed in previous lessons about sampling design, determining the number of sampling units as well as number of points is something that needs to be determined. Most often, this is best determined by pilot sampling. In general, when we are doing sampling with points, we usually need to have several hundred points combined in order to be able to characterize the vegetation of the site. Obviously, determining the number of points depends on a large variety of factors. Now let's move on to line intercept methods. In this case, we are looking at the interception of canopy or basal vegetation along transects. Line intercept is best used when we have compact and very distinct plant canopies or plant bases. And similarly to points, the way that we calculate canopy cover using line intercept methods is by dividing the length of the canopy intercepted by the transect length and then multiplying that by 100. As we see in this diagram that was originally published in Elzinga et al., essentially if you have a transect, what we look at is the distance of the intercept of a plant canopy along the transect. So let's look at a very simple example where we have plant canopy with a transect going through it. This is a 10-meter transect, and we can see in the orange lines there the length of the canopy where it's intercepted by the transect tape is in orange. If we take those individual lengths of intercept and add them together and then divide that by the length of the total transect, we find that the cover in this case, there was 6.1 meters of canopy that was intercepted, and divide that by the total length of the transect for 61% canopy cover. Now let's look at how this applies to basal cover. In this case, again, we have the orange intercept. We can see that in the case of this second plant here, even though the canopy is intercepted, the base of the plant is not, and so it's not counted. We only estimate the length of the intercept of the basal cover. Again, what we see is the total we combine the length of the intercepted plant base, divide that by the length of the transect, and that equals 10% basal cover. Now let's quickly look at what happens if we want to use to estimate cover from photographs rather than from field-based methods. And we can do this using either aerial images or ground-based photographs. We're going to focus on a program that was developed by the USDA Agricultural Research Service called Sample Point. And this is a very useful tool because the way that it works is you have an image. Your image can either be a ground-based image, as the one we see here, or an aerial-based image. And you can set the program to display a certain number of points, usually as a grid. We have a grid here of 5 by 5 in yellow. And the active point, the one that is being measured at this point, is in red. We can also specify a certain number of categories, whether they be functional groups such as grass, forb, shrub, cactus, etc., ground cover, litter, soil, rock, or unknown. And these categories can be set depending on what your sampling objectives are. If we take a close-up on that active point, we can see that it is on the canopy. But the thing that is really valuable with sample point is that we really are interested in the intersection of these two crosshairs. And we can continue to zoom until you feel very confident what is at the intersection of those two crosshairs. In this case, this image, this point, would be categorized as shrub because it is on the foliage of the snakeweed plant. So in conclusion, when we're using points and lines to estimate cover, there are a variety of approaches that are available. The decision about the approach that we use should always be guided by our management and sampling objectives and by a consideration of the site and species characteristics that we're working with.